So I love to do science. I feel very fortunate to have this job. I think that it should be an enjoyable activity, that humor contributes to joy. And so that's how I came up with this title. And it's to kind of represent the fact that I have a lot of interests. And so one of the challenges that I asked my students to do was to come up with clickbait titles for some of our research. And, guns don't kill people, aging kills people, right? This was actually a paper about aging, or habeas corpus for litigation data, right? You cannot get records of the law of courts. So we say habeas corpus, let's free it. So I want to kind of have some kind of humor about things. I want us to feel relaxed. I want you to feel able to interrupt me at any time. I want you to feel able to disagree with me, because sometimes I will climb on my soapbox, right? You'll imagine that there is a soapbox in here. I'll step up and I will start making these very emotional statements about the world, okay? And you can say, stop that shit. So, why do I say this about science? Because I'm interested in many things, and I am interested in the things that my students are interested in. So we have used stuff like bibliometric approaches to look which human genes get studied and which ones don't. We also looked at the effect of the speed at which things occur in a cell and the ability to carry out processes without mistakes, and why cellular processes are so complex. We also looked at what are the changes that happen in gene expression when we age. How we can look at the dynamics of a protein and figure out what's going on with the structure and its ability to change conformations. We also looked at ways to break documents into different topics in an unsupervised way, at getting rid of words that don't contain information about the content from text, how to build a more open and fair, just system by giving access to the data on litigation, and some very opinionated opinions about artificial intelligence. But today, I want to talk to you about two problems. And I will go relatively slowly over them, because I think one of the things that is very important for us as scientists is to think about what assumptions are we making implicitly that are driving our analysis and modeling, and are those assumptions justified? Okay. One of the most dangerous things is implicit assumptions, right? Because you kind of not control for them. So it's kind of very important to keep those in mind and having them front and center for you when you are attacking some question, some scientific question. And so I'm going to tell you about a case of one of these omics data sets called ChIP-seq. And so before that, let me start by kind of setting up the biology problem. So that's a photograph of James Watson. And at a faculty meeting, he may have said something along the lines of, now that we understand how genes work, we don't need things like ecology, as we'll be able to figure it all out from the genes. And, this is kind of based on what's called the central dogma, right? That in our cells we have DNA, that DNA gets replicated when the cell divides, that there is a process called transcription, that from the DNA copies into something called a messenger RNA. And that messenger RNA is then taken to a ribosome where it's translated into generating proteins. So if we understand what the genes are, and we understand this process, we know what proteins will be available in the cell, so we have understood all of biology. Good. This is all of biology. No, it's not. This is just metabolism, right? So there are things in here that are enzymes. They are catalyzing reactions. And that thing is sort of a network with many components, many parts. Okay, we know the central dogma, but accounting for all of these is kind of tricky. And this was a paper published in 2003, a protein interaction map of Drosophila melanogaster. Because these proteins is not like in our cells, it's protein as a function, and that function alone. So we need to do A, we use protein that does A. We need to do B, we use protein that does B. It's not like that. Those proteins interact. They change each other. Any biological process involves a ton of proteins. So, now I'm jumping on my soapbox. It's always very important for us as scientists to not kind of make someone into a god. So I like to remind people that James Watson was not only an unethical individual, he was also a racist and a misogynist. And besides that, he was wrong about this bullshit of the central dogma explaining all of biology. Right? So it's very important for us to realize that the people doing science are humans and they do mistakes and they are flaws. Right? So it's always good, I think, to remind people of that, because we have to kind of this idea, oh, that person was a genius. They may not have done any wrong. So, regulation of gene expression. Yes, the genes are encoded in the DNA. We make a transcription into RNA, and then we make the protein. But actually, that act of doing the transcription involves a ton of proteins that are coming together. And some of the things that are used is regions where some proteins called transcription factors will bind and make this process work smoothly or not. Right? So yes, we have the genes, we have mechanisms to transcribe them, but there are all these other proteins that are actually part of the process, regulating and modulating. And so one of the things that we would like to know is, for instance, which transcription factors, which are the little diamonds, interact with which target genes, the circles, and in which kind of cellular process, the squares. Right? Because the central dogma by itself is not very useful. If you do not have knowledge of this network of interactions, what can you do? Not much. So we would like to build this kind of networks. In particular, we would like to know which transcription factors modify the expression, the activity of which genes. And that's where ChIP-seq comes in. ChIP-seq is a method to enable us to kind of build this sort of data. Okay? For those of you that are not into biology, I hope that I'm not going too fast. If I am, what is your recourse? Oh, come on. Huh?
Ask.
Exactly. See? If an old person like Herman, right, that walks around with the walker and ask a question in a seminar, what about you young people that are in the fullness of youth, at your peak? Can't you ask some damn questions? This was a question. Can you answer it? Can you ask some questions?
Yeah.
Wow, not even a sound. Can you make a sound?
Yes.
Thank you. That is so great. So this is what we are trying to find out. ChIP-seq is a way to do it. And you start by having some sample of a cell where transcription factors are bind. Then you do something in which you really attach this transcription factor to the DNA very strongly. Then you use stuff to cut the DNA that is not bound to the transcription factor. Then you put tags to arrest it. Then you go and do sequencing of the DNA that you have. You amplify it, then you align to a genome, and there is all of this stuff that essentially comes with a curve like this. Okay? This means that in this position in the genome, there were a lot of things that we sequence. So there were a lot of transcription factors that were attached to this region, right? So what we do is along the genome, we end up with a curve like this. Okay? I'm not an experimentalist, right? Someone told me, please do not enter my lab because your hands break stuff. So I keep myself to a keyboard and a computer. And so we were interested in this part. There are errors and noise in all the other parts, but there is a part in which you use a computer to do matching to a genome to get this kind of profile. And then you are going to say, okay, there are some peaks in here, which means that a particular transcription factor, because these peaks are close to a gene, is interacting with that gene. Okay? So I say about implicit and explicit choices. During this process, there are choices that I can make. I can choose to which genome reference I'm going to do the matching. You know, the genome of any organism is not something frozen in time. It keeps being updated, right? So you could choose which version are you going to do it. Some people are so careless that they don't tell you which one they did. Annoying, because if you're trying to replicate the results, you can't. The other thing is this peak statistically significant or not? What about this one? Where do you draw the line? That's another parameter in your analysis. The last one is when you say, I found a peak, and you say, how far away from the gene is that peak? You can ask, is it so far away that it's likely not to have any significance? Right? Because the fact that something binds 1 million base pairs away from where a gene starts probably has no influence, right? So all of these things are parameters that you'll be able to specify, and you should do it in a principled way. So we looked at this study that had some of these networks observed for different organisms, and this was in the mouse. And here I'm showing you the places where the transcription factors are binding, those kinds of curves. So I said, there are these three things. This was done in the mouse. We compared the results in terms of how many peaks do we say we find based on 2 reference genomes for the mouse. And what you can see is that the peaks and the edges that you define in that graph, so these are box plots for different cell types, right? And you can see that actually the genome doesn't make very much difference. Okay? And the reason is that the areas close to the gene have been so carefully sequenced that those don't change very much. What about the p-value? So the important thing is to notice that this kind of light blue is 10 to the minus 5. And what you can see is that on a log scale, this thing doesn't change very much until you go past 10 to the minus 5, in which the number of peaks that you identify starts to explode. What does this mean? That you probably need to be strict enough not to kind of start saying that noisy signal, right? So what we say is that anything that is smaller than 10 to the minus 5 is a good parameter because these other things do not depend so much on it. Naturally, you should always do some robustness checking, but this as a guideline is good. Now, what about saying, oh, if a peak is farther away from the start of the gene than a certain distance, I'm going to ignore it. So we can change that from 0 to 10,000 base pairs in increments of 1,000. And you can see that this thing just keeps growing, 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 growing. And it's kind of, there is no explosion, but it's sort of, what's the right amount? And people have taken this intuitive idea to kind of choose whatever they want. And so these people used a certain threshold, we used a different one. They didn't report what threshold they used, and we compared what we found versus them. So there are here different cells, right? This is the different cell types. And if it is blue, only us found that peak. If it is purple, both of us found that peak. And if it is red, only that paper found that peak. And what you see is that there is no blue, right? There is a little bit in here. There is a fair amount of agreement, but then there are a huge number of things that only them found. So you could ask,
Is there any like ground truth here? Like, is there like another, like a way that we could determine if
that is a
like who's right?
beautiful question. That is a beautiful question. There is no ground truth, which makes it all the more challenging because you have to find a set of arguments and processes to decide if this is good or not.
Is it possible to do something where you kind of like remove the transcription factor from the environment and then?
It is, but then you get into low throughput stuff and you can only do it for a few of the things.
Okay.
So this is an important point, this one. Okay. So we looked at the red and we asked how far away from the start of the gene were they? This is a log 10 distance. So this is 10 to the 5. This is 100,000 base pairs. A lot of these peaks that we did not identify were located more than 100,000 base pairs from the start of the gene. I would argue that those are unlikely to be biologically relevant. You could ask, is there an explanation?
Isn't it possible that it would be an enhancer or a repressor that would act at a far distance? I mean, these things exist biologically.
It could. And this is the problem. So one of the things that we found out is that some of the things that were really far away, the explanation was that they had reported in a previous paper that that interaction exists, right? When you have something that is a flexible parameter, you can stretch it or compress it to find whatever you are looking for. So we need to do something different.
Wait, Luis. So you have your three hyperparameters that you're varying, right? I wanted to ask a technical question. You can imagine that your three hyperparameters, they're actually coupling as you vary both of them or three of them at the same time. Do you look into this at all?
No, because the three parameters are actually very independent from one another. So let's think about it from a first principle things. We have our genome, right? And how do the bindings for a transcription factor are just some sequence of base pairs. And this sequence, you can imagine, could arise by random mutation, right? And then if there are other mutations and insertions and deletions, you could imagine that this block is doing a random walk in space. Okay? So if you have this kind of unbiased random walk, if you have random emergency anywhere, what you expect is to find a uniform distribution of these things. These are not biological. These are just mutations that are neutral, that don't affect anything. Right? And when you think about it, biology is sticky, right? I mean, things just bind to one another, and the alphabet is not very large, it's only 4, so there aren't that many possibilities. Now imagine that you get one of these binding sequences close to a gene for which there is a benefit to have it there affecting the gene expression. Then you'd imagine that the diffusion of this sequence would no longer be random, but it would be biased. It would be attracted to the start of the gene, right? If you model this as a potential well, then what you get is that the distribution of such sequences is going to decay exponentially. Right? So essentially what we have here is 2 processes. One in which there is an exponential decay of likelihood of them occurring with distance to the gene, and one in which there is no dependence, it's all uniform. So this is what the data looks like if you start counting how many peaks do you find at the distance of some base pairs from a gene for several transcription factors and for 2 tissues. So it looks very simple. Goes fast and then goes linear. What does the model predict? Well, the model predicts that the exponential behavior goes fast and then the uniform thing just goes linear, right? Integrate the uniform function, you have something that grows linearly with distance. Qualitatively, they look very similar. So how does it work? Well, we can have the data for one transcription factor in one cell type. We can plot it in a different way in which we plot the observed value versus the fitted value from the model. And if the model is perfect, everything should lie on the diagonal, right? The model prediction is exactly the same as the data. Okay, so a transcription factor in a tissue for which the model works gives you a straight line. Now let's do the 15 transcription factors that we have for that cell type. Not bad. Well, let's do the transcription factors for the 15 for the different 5 cell lines. The model works very nicely. What does the model tell us? The model tells us that biologically relevant peaks are distributed at the distance from the starting of the gene that decays exponentially, right? So we actually can say that there is a point in which a peak found in here is more likely to come from the biologically relevant than from the spurious noisy one.
I have a question that reflects maybe my lack of knowledge of biology, but isn't there a directionality to the transcription? Shouldn't there be asymmetry about the gene?
So I thought the exact same thing. And so there are 2 answers to that question. The first one is that some transcription factors do not enhance copying, they suppress it, right? So then you would want to be right in the middle of the sequence of the gene. The other one is that this is a sequential process in which you are recruiting other proteins. So it could be that something is there to help recruit other things, and then it's released when the copying starts. That's my explanation right now. The simplest model is one in which the two things are the same, the bias on the two sides are the same, and it seems to work to explain the data very well.
But did you look for bias? Did you look
Yes.
for that? Okay.
So what? So we have a process that seems to say some binding peaks are biologically relevant and others aren't, and this is a choice. But how can we verify that? We would like to have an alternative method that at scale would tell us this is working. Now, the transcription factor in principle is there to help recruit all this other machinery that is actually going to transcribe the gene. In particular, it's recruiting something called RNA polymerase. And so what we can ask is, so there are some things that we say are biologically relevant targets and others that we say are spurious. And we could ask how much recruitment of the polymerase is it doing? If the ones that we say are biologically relevant, what we should find out is that they get a lot of recruitment of the transcription factor, and it's kind of, decaying from the center. If it's spurious, there shouldn't be much at all, and it shouldn't be kind of peaked at the center. And so this is what we find. For the ones that we said are for real, we find a lot of recruitment of the polymerase. So we say there is a place there for a transcription factor to bind, and because of that, we find a lot of the polymerase there, while in the other ones, we don't.
How do you know where the polymerase is?
It's a different experimental technique. It's basically the same thing. You have the sample, the DNA, the polymerase. You put something that actually sticks it together. Then you break the cell apart. You chop everything except the DNA that is protected by the polymerase, and then you sequence that. So you do something very similar. And the good thing is that this is high throughput too. You do it for the entire genome. Another thing is that we expect that if a transcription factor interacts with a particular gene, there will be correlations in their expression level. Right? If this is something that promotes expression, the more transcription factor you have, the more of that gene you should have too. Okay? So we just look at, is the expression level on a different type of data, RNA-seq, correlated or not? And what we find is that for the cases in which we were predicting these to be biologically relevant through edges, we find much higher levels of correlation than in the other case. Yes?
To get the correlation, do you have to do mutagenesis at the proposed binding sites?
No. So essentially what you use RNA-seq, and you say, I know that this transcription factor interacts with this gene, so I will look at many conditions, and I will see how the amount of the transcription factor that I find correlates with the amount of the mRNA of the gene that I'm looking at. And if the two of them are correlated, it means that one is impacting the other. Actually, we know because one is a transcription factor and the other one is not. We know that presumably it's a causal thing. It's not perfect because there are not only one transcription factor affecting one gene. So that's why we do not find 0 in here and 1 in there. But the difference is very significant, saying that, yes, we find evidence that these two things are more strongly connected than for the other ones. So in a sense, these are providing a sense of ground truth because they are completely independent data sets that what we find, the signals that we find in there corroborate the hypothesis that the transcription factor was interacting with it. Okay, so how am I with time?
Halfway through.
Halfway through. Okay, how's everybody feeling? Everybody awake? Doesn't need to stretch. Enough. Any questions about that part? So, just to summarize, what were the things that I was interested in here? Two things. Many times we don't have a ground truth, at least not at scale. Many times there are many things, choices that we make in an analysis that we do not report because we think that they may not be important. And many times we think that we should have the flexibility to choose something however we want. And I'm kind of pushing back on a lot of those. Because you can do the same thing as those other people and just stretch what is biologically relevant. So whatever you reported in the past appears to be confirmed by your new data. So it's good to have something that is objective, in which the decision is out of your hands, in a sense. Okay.
Can I actually ask, maybe it's a naive question. Do people share the code that they use to do these analyses? Because
oh, don't we wish that? I wish that they at least reported some of the basic parameters that they use. I mean, the student in trying to replicate that, she had no idea what genome reference model they used. So she had to try like 6 to see which ones give better matches. She didn't know what p-value they use. So she had to try all sorts of values to kind of, so those things in which we compare their results to ours was really with what we found to be the best agreement with what they reported. I mean, it's like weeks and weeks of work because someone decided that telling you what they did was not important. You can see that I have some strong feelings about this. So, now for something completely different with some of the exact same problems. So this is the work of Fei Hong, who's sitting out there, and I think this is really amazing work. So you all remember when COVID was really bad? This is a chart of patients with COVID in the intensive care unit for the United States. And you can see that the numbers in there were some really big numbers. And this is tough because people, physicians and nurses taking care of patients in the intensive care unit have to deal with a lot of stuff. So there are lots of machines that are monitoring and actuing on these patients in a hope to make them feel better. And the doctors have to make a ton of decisions. Do I change the medication? Do I change the ventilation level? Do I let things as it be? Can I still do something for this patient? Should I talk to the family and decide that, the best thing is to let the patient die in a peaceful way? So these are all kind of important things. And so you would think that with data, you could maybe help these doctors. And so there is something called electronic health records. If you go to a doctor and they give you a test or a vaccine or something, they probably will scan some bar, and this will record what happened into a computer system. This was developed mostly for billing. But actually, there is a lot of cool information in there. There is demographic information about the patient. There is vital signs like heart rate, temperature, blood pressure. There are severity scores about how well you are doing. There are clinical tests like chest x-rays that can be ordered and stuff like that. And all of these gets recorded. And you think, wow, this is cool. Look at all this playground that I have to play. Oh, for the playground. But it's not so easy, right? Temperature. Well, here this is measured in degrees Fahrenheit, right? So it goes from what? I don't know, 80 something, maybe even lower than that if you're kind of doing poorly, to 100 and something. Blood pressure. It's a completely different scale. Glucose in your blood. Well, yet another different scale. Are they all measured at exactly the same time every day? No. Some are measured several times a day. Others are measured every 10 days, a chest X-ray or something like that. So how do you integrate data that comes with such different units and such different frequencies of being acquired? It's not really the goal.
What is the goal of treating this data?
The goal of treating this data would be to help the physician know what the state of the patient is. And I'm glad that you ask. What do we believe that should come out of there? Should it be something that is like a continuous variable in which maybe here you are good, and as that number increases, you are more and more likely to die? Right? Some of the scores that physicians have developed are a little bit like this. They are a continuous. They aren't, but they are sort of aiming at the continuous. There is one scale that goes, and here you are good, here you are bad. But that approach is not working so well. Could it be that actually what you have is discrete states? There is a red state, there is a green state, there is an orange state, and maybe each one of those states has different characteristics. They have different prognosis. They have different disease origins. What is important is that we do not know. I do not know if this is true or if that is true. Right? This is the problem. What is the ground truth? I don't know. So what am I challenged with? Me meaning Fei Hong. What was Fei Hong challenged with? She had a bunch of data, all in different unities, with different frequencies of data collection, that she had to organize to identify something that she didn't know what it was. Was it this or that? Easy, right? How do you know you are making the right choices when you don't know what the right answer is? You start being very simple-minded, okay? So we had lots of patients from a couple of ICUs. Some of them went home, some of them died, some had intermediate outcomes. They changed in a variety of ways. We had records of them through their stay in the hospital. We have data from when they were in the ICU. And the first decision we made was, we are going to look at the patients day by day. Many of the things are probably not measured more than daily. So we are going to collapse all the measurements that we have from a given day, and we are going to say, we are going to build a vector for this patient that has all the information for that day for that patient. So this was the first choice. We could have chosen a longer time scale to aggregate things. That's not so good because you lose ability to distinguish. You could try to do it with shorter time intervals, but then you didn't have enough data. So there is always a kind of trade-off between those things. Okay? So this is the first big decision that we made. What was the second big decision? The second big decision was that, so this is a signal. I think this is the kind of oxygenation of the blood. And what you can see is that when you have yellow here, you have measurements. When you have gray, you don't. And there are two things. There is a lot of gray, right? There is worse than that. The gray is not spread randomly around, right? It's good when missing stuff is random. It's bad when it's not. There's much more stuff missing at the end, and there are some patients for which you never see anything, right? So it's not like you can just go and assume, oh, everything is random and well distributed and stuff. It's not. And so the completeness of different things also changes. And you can see that for some clinical tests, you have very little data, even on a daily basis. So to summarize, what are some of the choices that we have to make? We have to make the choice at what time scale should we consider the data. We quickly decided that 1 day was good. What data should we consider? Because some of them are missing, and some are missing much more than others. Because we have all sorts of different data, we will have to normalize them. How do we normalize them? If we are missing data, should we try to guess what the values of the missing data are or not? And we have, let's say, order 100 variables in there. Okay? Now think about 100-dimensional space. And we had about 600 patients, maybe 20 days per patient. So let's say 12,000 data points. How well do you fill a 100-dimensional space with 12,000 points? You should be able to answer this with confidence. How do you fill it? Extremely well or poorly? Please? Poorly or extremely well? Huh? Do I have to prostrate myself? Very, very poorly, right? So we need to do dimensionality reduction, and we have to figure out which way to do it and how. You guys are really a tough audience. Yeah, I feel so bad. So the challenge, if we had some ground truth, then we could determine what are good choices, but we don't even know if there are clinical states or not. So that's tough. What is the ground truth?
Somebody survived. It's one thing. Okay. Somebody survived.
Another old person that is talking.
I have to complete my sentence. If somebody survived your calculation.
As Volodya was saying, there is an obvious truth. If you survive and sent home, you are probably very different from the person that died. Now, it's not that you are always very different. Two patients could have looked very similar here, and then one has a bad thing happening and goes to die, another doesn't. So we cannot look at all the values. What we should look is at the days very close to when they died or when they are discharged, right? Because those are the set of values that we should expect to relate to very different states. It's obvious, right? Someone that is about to die and someone that is about to send home should look different in our data. If they don't, whatever we are doing is pretty stupid. Right? So it's simple-minded, but it actually gives you an entry into a ground truth. We can look at all the choices that we made and figure out how different people that died are from the ones that were sent home. And the more different they are, the better. So we use something called the silhouette score of coefficient, which is essentially related to the average distance between points inside a group versus the distance to points in a different group. And you can look at things like what different normalization you use and what different types of variables you include, and kind of make your choices based on that. And you can do it for different databases. Okay? So even though in the beginning we didn't know anything, we can say these choices are terrible, right? If we don't normalize the data at all, right, then we get horrible, horrible results, right? So when you put in some vector temperature, non-normalized, and you have something else that is blood pressure non-normalized, and you are putting that in some space, things just don't look very good. So that gives us a mechanism to kind of decide which features to include, which interpretation strategy to use. But we still have to sort of figure out how to do the dimensionality reduction. Now, we wanted to do a method in which we have control, so we used PCA because we know that transformations are linear. We need to figure out how many principal components to retain and do they make sense. So we actually just varied the number of principal components that we used. We looked how much of the variability of the data they capture. And we also looked at building it from different sets to see were we finding robust results or not. In the end, we found that 4 principal components looking at failure, organ failure scores, and vitals were the best approach. And now we can plot all our data, right? So we have defined an embedding system, right? We can transform any day into using this transformation and put it in the common space. So now it's no longer just before dying and before going home. This is all the data. And you can project on different sets of 2 dimensions your data, right? Because you have 4 principal components, you have each pair. And what I want you to see in here is that that thing here is not uniform, right? So the dots seem to congregate around some areas of space. That suggests that there are clusters. That suggests that there are discrete clinical states. That is not a continuous. There are some attractors is the wrong word, but there are some kind of combinations of these features that clump in space. So the task is now to find them in 5 dimensions, in 4 dimensions. So we know that it's not continuums, it appears to be discrete states. How do we find the clusters? Okay, you could say you could use k-means, hierarchical cluster, Gaussian mixture models, and what you find out is that all of them work beautifully in those little examples that you find when the model, when those approaches are being illustrated. As long as your little data is perfect. It doesn't work. So we actually, I'm using the royal we, Fei Hong did all of this stuff. She found this density peak clustering because one of the things that you find in those methods is that many times they tell you a center of a cluster is in a place where there are almost no points. This makes no sense, right? This one forces the centers of cluster to be where you have high density. But what we found out is that if you just run it once, there is too much noise. So you actually should use an ensemble approach. And so we introduced what we call ensemble density peak clustering. And what you find out, and I'm showing here just a projection on 2 dimensions, is that when you bootstrap your data and repeat the process, you find a bunch of centers in here, a bunch in there, there, and there. And then this is the kind of nice data that you can use k-means to find the clusters. Because it's nicely separated, not very many. All of them have about the same number of points. All the conditions are. So we end up finding 5 clusters. And this is the final days of them. And this is all the points of them in there. And what do we learn? So again, what's the ground truth? Right? I mean, it's like, yeah, we developed this very sophisticated machine to make sausages out of a bunch of meat. Are these good sausages? Are these chorizo from Blackfoot pig, Iberian Blackfoot pig? Or is just some crap from a factory farm with pigs that, right? There's a difference. So you have to compare what you are doing to something else. So you can look at the scores that are traditionally used. And we just separate these scores into quintiles. Okay? So the idea is this traditional score that critical care physicians use, the value as it increases, mortality risk increases. Okay? So what do you find? The lowest values, and the next one, actually there isn't much mortality difference, but to the highest values, there is a high mortality risk, right? So the chance of you dying, if you are classified in this group, is 7 times as large as if you are classified in that group. Okay? Not bad. 7 times. That's a good discrimination. Doesn't seem that these two have very much difference. So what do we do if we look at our clusters? So we actually have 3 clusters that are very much related to mortality. Cluster C1, C2, and C5. C5 is the one in which you are more likely to die. C1 is the one that you are least likely to die. On a 24-hour window, none of the patients that we ever classified in C1 died. About 1% of the ones in that lowest quintile died. Okay? So this is an infinite game. Yeah, just kidding. But let's look at these and these. There is a factor of 7. Here, there is a factor of 31.5. If we say that you are in cluster C5, it's really bad. There is a 7.5% chance that you are going to die within 24 hours.
So what's your suggestion?
My suggestion is that you should talk to the family. No, jokes aside, it's like at some point you should know that there is nothing you should do. The patients that get in here actually do not leave here. So even if they don't die right away, they remain there. And this is actually a good thing to know, because at some point you should realize there is nothing left to do. So this was on the train data. You can see that you find similar kind of things for the test data and also for a different database. So it's kind of robust across data. And so this is actually meaning that you are actually discriminating the outcome. You know how well or not well the patient is. Now, the other 2 clusters were actually related.
Just a quick question. I'm wondering, because you're comparing to quintiles, which I guess you have to have 20%. So I'm wondering how big these clusters are.
Yes, so we chose quintiles because that's when you get the best match to the values for our clusters. So the clusters are kind of well represented in that.
So to do this, you have this data set that the doctors are collecting. Were you ever able to show that like some things that you're measuring are not important to the results at all, and you can remove those?
We haven't started to doing that. So if you go back to when we look at the loadings in the PCA, you can see that there is a small number of variables that have kind of a bigger impact. And others are actually always very extremely correlated. So there are certain things that would make more sense to focus on. Another thing that we haven't looked at, but it would be interesting, is to see, because we also have treatment, so we know some drug was given to someone. So you could say, treating the patient in this way, actually to which other cluster did it move them? But it's like, this was already a lot of work. So some data that I'm not showing here is related to the etiology of the patients in each cluster. And this is kind of the origin of the pneumonia. So physicians usually divide pneumonia into community-acquired, hospital-acquired, and ventilator-acquired. Actually, we didn't find very much correlation at all of those things in the States. What we found a strong correlation was between viral and non-viral. And because a lot of these patients were during COVID, in particular about COVID and no non-COVID. So that led us to ask the question, could some of these other clusters be related to the origin of the pneumonia? And so what we look at here is the rate at which the patients classified even a given cluster have COVID or not. And Q3 and Q4 in here have no meaning, right? We should find no difference, right? Because it has nothing to do with that. So it's here not to say, oh, look, we do better, but just to show if you select things at random, you would get about the same rate, right? So here I'm not saying, oh, look, we are doing so much better. It's like, this is the expectation if you were guessing. And this is what we get. And what we get is that the patients classified in C4 have a much higher rate of COVID than the patients classified in C3. So this cluster is the cluster of patients that came in with COVID. So it's actually telling us about what's going on. Actually, the patients get into that cluster before we have a COVID diagnosis in the system. Which, when you think about it, in the beginning, when we didn't have tests, this was pretty good, right? If you had said all of these patients are in the same cluster, you could have said there is something in there. This is not a flu patient. This is a COVID patient. So these states, in a sense, are picking up something that is important about what's going on with these patients. And in principle, they will give us more homogeneous groups to study the impact of treatments. So this is kind of a summarizing. One thing that I'm not addressing here is the fact that we have to make decisions to integrate data from two very different databases. In particular, this one, Mimic, didn't have any COVID patients because it occurred before the COVID pandemic. And so one of the big clusters that you found could not even be there because there were no COVID patients. So that was kind of interesting, and you can read more about it if you are interested. And so I want to thank you all very much. Actually, I don't want to thank you. I'm just wanting to thank the faculty that actually asked some questions. You guys were miserable. No, not everybody was miserable. Most of you were miserable. I have to say, very disappointing. They at least laughed at all my jokes, but some of them laughed at my jokes. Anyway, if you have any questions, I'll be more than happy to answer.
Was there any effect on like the timeline of data you included for these decision making or for the clustering? So like you said, like you were including only data around the end of the.
Oh, that was just to evaluate the choices of which features to include, right? It's sort of, or for instance, imputation is a big thing. Right? So you have a missing value and you could say, how do I try to guess the value that is missing? And there are different approaches. You could say, I'll just put a 0, or I'll just use some complicated model and make a linear regression on the days around and replace with that other value. What we found out is that if we just carry over the previous value from the previous day, that worked pretty well. it only worked well if you did it up to 2 days. If you kept doing it, then actually your guess was no longer good. So it was used to make those decisions. When we were looking for the clusters, we put all the data in.
Okay.
I actually have a follow-up, which is, but how do you know that the features that are at the end are the features that are actually, like, is it possible that you could have a set of features in the, if you look at the time window beforehand that are better at predicting, but the
Yes.
features that are actually predictive are going to do at that end window?
Yes, that is an excellent question, and there is no answer to it, right? You could start doing something like that if you were trying to say, I have some other ground truth, and I'm trying to optimize with regard to that. And in hindsight, for this database, we could have used COVID-19 as another kind of target measure, because that would have actually been valid at any point, right? It's sort of, you could ask, what are the features that at any point give you the most information about the patient having COVID or not? The point is that for you to kind of not do wishful thinking, you need to have something that you can grab and say, I can trust this information. And so COVID is potentially one of them, but yeah.
Just to follow up here, it depends on the pathology of the disease. For some, you have like a stochastic distribution of where you would die. Like if I have a very weak heart, like a heart failure or something like that, that would not be predicted right before, right? That would be the presence of that transition into the disease. That's very simple.
And so some of the things, for instance, that turn out to be surprising is that you'd imagine that if you look at it in another way, age is very predictive of stuff, and gender too. So older men are more likely to die than somewhat younger women. But actually, when you are looking in this framework, it just, if all your physiological signs get into this way, age and gender not providing you any additional information. So it's kind of, it's a limitation of not having a ground truth, but the physicians themselves don't know about these other things. And so identifying situations in which you can have a group that maybe there would be some way to objectively and using different information identify would be the way to do it.
How would this be applied to, say, more current data where COVID is not so prevalent?
Oh, it's just completely fine. So so we identify these set of variables, and if a patient does not have COVID, we would hope that it's not classified in the COVID cluster. Now, people are talking a lot about the new bird flu and stuff like that. Would we be able to see that? Potentially, if you add those patients with those characteristics here, the clusters that you would identify would change, right? Because this would be another group of patient days that share some characteristics, but that are occupying, hopefully, a region of space that was left mostly open before. But this is one of the things, is that what we found out is that it's actually better to have less data with more variety of outcomes and conditions that have more data that is not as rich. So when we look just at the mimic data, there are a bunch of things that we don't see. So most of the patients in the mimic data actually survived. The other thing is that there was no COVID. So it's kind of, you don't see some of these structures emerging that you see in the other data. So you want, it's not only that more data is better, because if some of the things that you are looking for are now very poorly represented, it becomes very hard to see them because the other things are kind of dominating what you observe.
So obviously not at this point, but is the eventual goal of developing these types of data analysis, would it be to be used to say evaluate the say success of certain interventions, or as a like doctors dealing with patients in a hospital, what to do at that moment? Like, is it a kind of would be used in hospitals or more just evaluating?
So I see it in two ways, and one of the goals would be to add some, and this is an example. We have a few other things that we have been doing in which we are using electronic health records to actually help physicians with diagnosis or kind of treatment plans and things like that. And it would be really nice, and there is nothing that would prevent you from pulling the data. Well, Epic will prevent you from pulling the data. That's the people that sell the thing. But if you could pull the data and calculate it in real time, you could return this information to the physician to help them with decision making. And so that would be a direct benefit, because sometimes physicians miss certain things. The other one is sort of when you are trying to look at the impact of, for instance, a treatment option. You want to be able to partition your data in intelligent groups, right? And so, you know, it was like in medicine for the long time, they look to men for participants in clinical studies, even for things that would be related to women's health. Right? So men were used as the model for breast cancer. Right? It's kind of, you are using a bad model to do an analysis. People didn't look at race, right? But there are some conditions that Ashkenazi Jews are more likely to have, are some that African Americans are more likely to have. And so when you are looking at an intervention, if you have very few people from these groups, and you have a lot of people that are not, it could be very highly effective in the group. But because you have all the other people, you wash away the result. So the question is, how do you build these intelligent groups that you expect to have more in common so you actually can analyze them separately? And so this would be the thing, right? You could look at, if you look at the treatment for C4 and C3, it could be a wash. It actually could be that it makes one group better and another group worse, right? And so by being able to carve that and the impact, you are gaining the ability to see things that otherwise you wouldn't. So I think that there is potential in both aspects of it. It's also, I think there is potential in the thinking and approach to look at other conditions in the ICU that are just not pneumonia. This was focusing just on patients with pneumonia, which is a very important condition, but it's not everything.
I think we've run out of time, so let's thank Luis again.